And Michael is renowned for his work on the genetics of adaptation and speciation using genomic tools and population genetic methods, primarily with mammals, but not exclusively. And that's all I'm going to say, because I want him to have as much time as possible to tell us about the genetics of adaptive melanism in pocket mice.
Great, thank you so much, Jim. You know, for people who are standing, there's a chair here and there's two here. So come in if you want to. Don't be shy. There's still two chairs here. Well, thanks so much. It's really just great to be here. I am getting a little tired. It's the third day and just talking to people, and so. But I just had a little caffeine, so it should be good to go. It's really fun to talk to everybody here. It's just been great, nonstop. The science is tremendous at this place. I've enjoyed meeting with all the students, postdocs. The interview should be 5 days long. I get to really talk to everybody. I'd be dead at the end. So I told you on Monday that there are two, that I like to think of evolution as consisting of anagenesis and cladogenesis. And I talked on Monday about the genetic basis of speciation and how reproductive isolation arises. So today I want to talk about the other major area that I've been interested in for a number of years, which is the genetic basis of changes within lineages, and in particular adaptive changes. And as you'll see, we have been focusing, or what I'm going to talk about today mainly is about a particular model that I think has been very helpful in understanding some general principles of the genetic basis of adaptation. I guess I want to begin by saying that, as you all know, it's been 2 years now since the origin of species. There were many symposia. In fact, there was one here on the anniversary of the origin. And after 150 years later, we know a lot about variation, a lot about selection, two of the three ingredients for evolution by natural selection, but relatively little about the genes underlying adaptation. And this has been a little bit of a tough nut to crack. And I guess there are a number of approaches one could take in principle to trying to find the genes that underlie adaptation. One that's become very popular in the last 10 years, and one that we have used and published some early papers on, is to scan the genome in some way look for signatures of selection. And the idea here is you're agnostic with respect to phenotype. You look across the genome. It's not a new idea. There were Alzheimer's studies that did this, approaches going back to the 70s. But the idea is you look somehow across a proportion of the genome. You look for a signature of selection. And then once you have that, either at a gene or a region, you try to make links to the phenotype, and then ideally to understand how that phenotype might affect fitness in a particular ecological context. There have been many, many scans of selection in lots of different organisms. There's now just a reasonably sized literature on this, probably greatest in humans. And in looking over that literature, I think there's one thing, just thinking about humans as an example, that's consistent, and what's consistent is the lack of consistency. If you take all of the studies that have done genome-wide scans for selection in humans, and you simply lay them on top of each other and ask, are the same genes and the same regions identified, the answer by and large is no. Josh Akey pointed this out in a really nice review paper a few years ago. Different studies identify different regions. Now, there are a whole bunch of reasons why this might be the case. The methods that are used are often somewhat different, and they have different power and different situations. But I'd like to think that if we're really detecting a consistent signature of selection, we might get more overlap than we do. The other thing that I would say, the other general conclusion that comes from this kind of approach is that very few studies have made the complete link from genome scan to ecological context. There have been so many scans for selection, but you can count on one hand, and maybe, depending on how you define it, one or two fingers, the studies that have actually found a region that was not predicted or expected a priori, and then figured out what exactly has been under selection in some ecological context. One of the best examples of this was published in PNAS about 5 years ago by David Begun, where they were doing a scan in Drosophila simulans, and found a region they were not looking for, not expected, that seemed to underlie insecticide resistance. There are lots of cases where people have found signals that for which we already had a candidate, like lactase persistence in humans, but that's different. That's having a candidate in mind. So, as interesting as this approach is, and I think it does reveal a lot, I think it's been tough to use it as a way of making links between genotype and phenotype. So the question is, how do we get beyond scans for selection? And one obvious answer is just to do the reverse. That is to start with an ecological context that we are interested in and for which we have some understanding of how selection might operate and why different phenotypes might have different fitnesses. So we can make this connection down to phenotype, but then try to get to the gene from the phenotype. And this can be done in cases where we have a good set of candidate genes, and I think now, even in cases where we don't. And so that's what I want to talk about today. So for the first part, really most of what I'm going to talk about is work we've done over the last almost 10 years now on adaptive melanism in pocket mice. Some of this is older work, some of it is new. And then I want to take just 5 minutes at the end and tell you about some new work where we're not using candidate genes to look for adaptation to extreme environments in house mice. And both of these projects involve lots of field work in lots of neat different places. So, but the second thing is really more of a future directions topic. Well, what are the things that we really don't know about the genetics of adaptation? I gave you this list on Monday of things that I think we don't know about the genetics of speciation. This isn't a very different list. I think we'd like to know how many genes are involved, whether they are single genes of major effect or many genes of small effect. Fisher had very strong ideas about this. How important is dominance? Haldane argued that while beneficial mutations are equally likely to be dominant or recessive, those that get fixed are more likely to be dominant. This is sometimes referred to as Haldane's sieve. But we don't have a good general test of that. Are changes due to changes in protein structure or gene regulation? There's been a very lively debate over the last 5 years involving people like Greg Ray and Sean Carroll and Jerry Coyne and Hopi Hoekstra about this. It's not a new debate. It goes back many years. What kinds of mutations? Is adaptive evolution caused by point substitutions, caused by this supposed to be transposable elements, or insertion deletions? So insertion of transposable elements may give rise to adaptation. What kinds of genes are involved? And are there different genetic solutions to the same evolutionary problem? That is, if you have a similar selection pressure, do we often see similar genetic solutions to that? Well, I think one way to get at this, and not the only way, but one way is to start with a simple trait with clear ecological relevance for which we know something about the molecular details. And I've argued now for a number of years that coat color variation is a good example of that. So this is the rock pocket mouse, Chaetodipus intermedius. It's a Sonoran desert heteromyid rodent that looks like this in most of its range. That is, it's light colored and it lives mostly on light colored rocks. It's restricted to rocky situations. There are a number of lava flows across the southwest with rocks that are sometimes very dark in color. And on those rocks, on those dark rocks, you see these populations of mice that are very dark. This is an old story in mammalogy, and I can't say this enough, especially to the students here. There's so much value in reading the older literature. I got the idea to start this work by reading old papers from the 20s and 30s and 40s by Lee Dice and others. So this was well described decades ago and was just sort of ripe for looking at the genetic basis. Mice that do not match their background stick out and are probably removed by predation. Owls are probably major predators, and you might think, well, what difference does it make? Owls are out at nighttime. The mice are mostly nocturnal. And Lee Dice did some beautiful experiments. He did an experiment in 1947, actually with Peromyscus, not with these mice, but with Peromyscus that also vary in color. And he did these in large rooms with barn owls. He actually used 2 different species of owls, but most of the work was done with barn owls, and released mice in this enclosure on light and dark backgrounds and measured predation, and he did it at different light conditions. The interesting thing is, if you have no light at all, so in a totally darkened room, if there's no structure on the floor of the room, the owls are actually pretty good at catching a mouse, so basically without eyesight. And so you might think, well, so it doesn't matter. It turns out the owls are not very good at doing it when there's any structure for the mice to hide. And given very low light conditions meant to simulate the kind of light that would be outside at nighttime, the owls discriminate to a large degree based on colors. So that is the basis for thinking that this might in fact be adaptation for crypsis. When we started this work, we had this list of candidate genes. That is, there were about 100 known loci that had been implicated in color variation, mostly from work in laboratory mice, but also in other mammals. And of those, maybe 15 or 20 had been cloned and molecularly characterized. And that is why we picked this phenotype, and this list has grown in the last 10 years. So here's a picture of Dice, who is at the Ruthven Museum of Zoology. And there are very large collections of mice known as the Dice mice. He did a lot of breeding experiments. And this was one of the key papers. This was from 1937 by Dice and Blossom, Studies of Mammalian Ecology in Southwestern North America with special attention to the colors of desert mammals. And what he showed in this is if you just go out and measure reflectance off rocks from a lot of different areas, these have high reflectance. The Tinajas Altas mountains are very, very light in color. And the Tularosa lava beds, they are very, very dark. So there's this wide range of variation in substrate color. And then he went and measured reflectance off of mice caught. These are pocket mice from these areas. And you see this similar variation in color. And he showed, in fact, that they were highly correlated across geography. We focused primarily on these four populations. Here, the range of Chaetodipus intermedius is shown down in the bottom right. So Tucson is well situated for working on these mice. But as I was telling some students, this is part of the Cabeza Prieta National Wildlife Refuge. The main caldera for this volcano is in Mexico, but it gets into Arizona just a little bit. And then the Very Goldwater bombing range is basically just north of there. And so to get to this field site, we have to call up the Air Force and ask permission to cross. And, but as a result, the wildlife refuge is underutilized, and the habitat is spectacular. When we catch mice from these different lava flows, we see this kind of pattern. So these are pairs of mice from these four localities, dark mice caught on the lava flows, and light mice in each case caught just a few kilometers off of the lava flow. And what jumps out at you looking at this is that the mice from the different lava flows look, at least superficially, quite similar to one another, raising the possibility that maybe this has evolved independently a number of times. And this is one of the questions that we've been interested in. This is just to give you a feel for what the habitat looks like. This is in the Pinacates. This is actually a road cut here. And you can see just a little bit of the light soil underneath, getting underneath the spray of volcanic ash there. This is Carrizosa, New Mexico, from a Google Earth image. And the caldera was up here in the north, and it flowed down like this. This is one of the younger lava flows in North America. It's about 1,000 years old. At its narrowest point, it's not more than a couple of miles wide. The White Sands area is at the south, and then as well as further south from here. So it's a very dramatic landscape, and there's just an absolute sharp line dividing the light rocks from the dark rocks. And as I'll tell you at the end of the talk, I think that coarseness of the environment, that abrupt transition, actually affects the genetic architecture of traits involved in adaptation. And here you can see it very, very closely. So a mouse can simply walk from a very light area to a very, very dark area. This is campground that's actually on a hill, and you can see how the lava flowed around it as it went down this valley. Okay, I thought, at the risk of taking a little time, I was just going to show a little bit of a movie with the, if I can figure out how to do this. So there was a, I'm just going to leave it like that and do this without sound. So what you'll see in just a second are some pictures of the lava flow from the air, and then the mice, living mice on the lava flow. This is a film that was made as part of a PBS Nova special on evolution for the Darwin anniversary, and but it gives you a feel for the habitat, and we'll come up to the lava flow. This is in Carrizosa, so it's basically this lava flow down here, and this has now been made into a film by the Howard Hughes Medical Institute, which is being distributed to high school classrooms around the country for the teaching of evolution, as an example. I'm just going to show you another 30 seconds or so, but I'm showing it partly because there's some great footage, and also because some nice pictures of the mice, and you can see how they really stand out. We have these great predation shots, which of course were fully staged. This is just glitzy stuff for this. But that's Sean Carroll. I'll stop there. I think there's some more pictures of the mice.
He kind of sounds like him.
There you can see the edge of the lava flow. All right. The other reason I show it is because I think it's an example of the kind of outreach we can all be doing. This is meant to help educate high school students around the country about evolution, and I think museums have a particularly powerful role to play in this sort of thing. Okay, so here are the 4 lava flows, the color of the substrates on which we've caught mice, and the proportions of light and dark mice that were caught on each of those in each of those areas. And I'm showing this to you so that you get the feeling that if you go to dark rocks, mostly you catch dark mice, but you do get some mismatched animals. And conversely, if you go to adjacent light rocks a couple of kilometers off the lava flow, mostly you get light mice, but you do get a few dark animals. And we think that there's migration back and forth. The lava flows differ in age and size as well. Okay, so in thinking about the genetics of this, we had to dig into the molecular details and the developmental biology of how pigment is produced in mammals. And one of the key things to understand is that if you take a single hair from the dorsum of most mice, so this is sort of a wild-type phenotype, an individual hair has this banded pattern with a dark tip, a light midsection, and a dark base. Pigmentation in mammals there are 2 basic kinds of pigment, eumelanin and pheomelanin, produced in organelles called eumelanosomes and pheomelanosomes. And the control over which of these is produced is under a couple of key interacting players. So alpha melanocyte stimulating hormone is a peptide hormone that binds to melanocortin-1 receptor, which is a cell surface receptor, which when activated increases intracellular levels of cyclic AMP. This is a signaling pathway that leads to the production of eumelanin. When agouti is produced, this is a protein which is an antagonist, together with mahogany, binds to MC1R and shuts down this receptor, resulting in the production of pheomelanin. And so we knew this going when we started the work, and we also knew that one of the key differences between the light and the dark mice was in this banding pattern. So if you look on the dorsum of a light mouse, you see these banded hairs, whereas on the dorsum of a dark mouse, you see unbanded hairs. It turns out that band in the middle of the hair is caused by a pulse of agouti expression as the hair is growing out. So it's really this sort of intricate control between the growth of an individual hair and the expression of proteins in the hair follicle. And so because of that, we thought that either agouti or alpha MSH or mahogany or MC1R were reasonable candidates for this difference. I should also say that if you look on the bellies of both of these mice, they're light. And they're light not because they don't have any pigment, but because they have a pheomelanin produced. And because of that, we believe that the agouti protein is produced and it's functional and is expressed constitutively on the bellies of mice, resulting in all white undersides. And that'll become important later. From laboratory mice, we knew that you could take either MC1R or agouti and identify an allelic series that creates a dominance hierarchy. So the darkest alleles at MC1R are dominant over successively lighter alleles, and the reverse is true at agouti. And this can be understood by thinking of dominant alleles in both cases as corresponding to gain of biochemical function. So when MC1R is most active, eumelanin is produced, and so gain of functions make you darker. Conversely, agouti is an antagonist of that, and so gain of function mutations in agouti make you most light. And this too will become important when I show you some breeding experiments that we've done. So the approach was very simple. We thought we would try to take SNPs in candidate genes and look for associations between genotype and phenotype, with the idea that if we find, imagine for example that this is our marker, our SNP, we find a perfect association, then there might be a causative mutation somewhere nearby. And I should say, pocket mice are completely uncharacterized genetically when we started this at the nucleotide level, and so we really had to rely on candidate genes. We identified SNPs in a number of candidate genes using a mouse and human orthologs, and didn't see associations between color and SNPs in most candidate genes. And we looked at the 4 that I mentioned as well as 5 or 6 others. And initially we were focused on one population. And in this population, it wasn't not the first gene that we looked at. We did find eventually an association between MC1R and color phenotype. And we sequenced the complete gene. And that was done now 8 years ago. And I'm going to go through this very quickly because I want to get to the newer stuff. But I will say that this is what the association looked like. There were 4 non-synonymous changes in the gene that were in complete linkage disequilibrium with one another. And the association was perfect. So all light animals were homozygous for these 4 residues. The heterozygotes or the alternative homozygotes were all dark. And that was consistent with what we knew about the dominance hierarchy. So heterozygotes and one homozygote are dark, whereas the light allele seems to be recessive. And we also found that all 4 of these changes result in changes of charge. So they were the kind of mutations that looked like they might actually be functionally important. Furthermore, when we looked at patterns of genetic variation among what we're now calling the light alleles and the dark alleles, without doing any fancy analysis, you can just look at this and see there's very little variation among these alleles, much more among these, and so the idea is if one of these has been positively selected and quickly swept up in frequency in a population, it would reduce variation, and that's in fact what we saw. The final test, though, was to show that the receptor actually functioned differently, so we cloned the receptor into an expression vector and transfected cells in culture. You can do this in culture human kidney cells, and actually a mouse receptor, a pocket mouse receptor will assemble properly, and then you can target it with the normal peptide hormone that you can buy off the shelf from Sigma, and measure intracellular levels of cyclic AMP. And when we did this, we found, in fact, that the dark allele had higher activity over a range of concentrations of the normal ligand compared to the light allele. And this is exactly what you would predict if if these alleles were in fact functionally different. So I say here ongoing work using site-directed mutagenesis to test the effects of each of the mutations separately and in various combinations. Remember I told you there are 4 mutations there. This is something I would love to have a student be interested in pursuing. Additionally, if you look at the level of variation among the dark alleles, so as I showed you a minute ago, it's reduced. This is within the gene itself. As you go immediately upstream or downstream from the gene, levels of variation climb back up. as if selection is acted just on this gene, and that further provides evidence that this is the target of selection. If you look among the light alleles, you don't see that same sort of pattern. If you look at patterns of linkage disequilibrium, we've done this for many alleles. I'm showing you just a subset here. This is the gene. This is the upstream region. That's the downstream region. Here are the 4 non-synonymous substitutions. There's a 5th substitution that's a synonymous site that's in complete linkage disequilibrium. But what you see is if you just go upstream a little bit, the association decays very rapidly. And so the idea here is that if there was a site at an adjacent gene or far upstream or far downstream that in fact was the causative site, and you just happened to be finding these non-synonymous substitutions that were in linkage disequilibrium with it, then you might expect, if that were going on, then you would expect linkage disequilibrium to extend. Because it doesn't, again, that further establishes that it's really this gene. We can also, from the decay of these non-random associations, estimate the age of the allele. And we've done that and concluded that the most recent fixation was somewhere between 35,000 and 120,000 years ago. So it's a wide confidence interval. The lava flow is about 1 million years old. And this is the age of the most recent fixation. So there are 4 mutations that I showed you. It may be that the first one arose 1 million years ago or 900,000 years ago, and that there were some, those other 3 came later. This allows us to estimate the age of the most recent one. So this is all published work and is what I view as very strong evidence that MC1R causes dark color in this population. The non-random association, the dominance patterns follow the predictions, the mutations result in charge-changing substitutions. I didn't talk about this. The 4 presumptive functional sites are conserved across all other species of pocket mice. We see a signature of selection. We've done functional tests to show that the dark allele encodes a hyperactive receptor, and the association is limited just to the gene. It decays upstream and downstream. This may seem like overkill, belaboring the point that this gene is really involved in this. When we published this was one of the first examples of the specific adaptive basis of a phenotype in a natural population. We really wanted to establish beyond any shadow of a doubt that's what was going on. And let me just say a couple of words about how strong selection is, and then I'll tell you from that. I think there's been a lot of subsequent work that sort of extended this result on this and other species. Once we had the gene in hand, we could then begin to estimate selection coefficients with these alleles that we believe underlie this adaptation. So we sampled across this lava flow, and this is work that was done by Hopi Hoekstra when she was a postdoc in my lab. She sampled across this lava flow from light rocks onto dark rocks and then back onto light rocks, and caught both dark and light mice. And what you can see is that dark mice were most common on the lava flow, but there were some light mice that were caught off of the lava flow. And then she looked at the frequency of mitochondrial DNA going across this transect. Mitochondrial DNA being a presumably neutral marker, or at least not subject to selection for crypsis. And what she found was that there was essentially no difference in frequency, consistent with the idea that there was a lot of gene flow. However, when you go to light rocks, you see some dark alleles on the light rocks on both sides of the lava flow, and you also see some light alleles on the dark rocks. That is, the frequency of this dark allele does not go to 1 and to 0. There's some mismatch that occurred. And so we reasoned that one could use a very simple population genetic model of migration selection balance to estimate the strength of selection. If you infer that this frequency, that is the frequency of a dark allele on light rocks, is a balance between 2 things, one being the introduction of that allele from migration, and the other being its removal by selection, then if you can estimate the rate of introduction, the migration rate, by some other method, you should be able to estimate the strength of selection. The mitochondrial DNA provided a way for us to estimate the rate of migration. And so that was the logic behind this work, and this is what Hopi found. Using a simple migration selection balance model, and our estimates of migration derived from the mitochondrial work, selection against dark mice on light rocks was estimated to be lower than selection against light mice on dark rocks by maybe an order of magnitude. So, you know, depending on your view of evolution, you can think about whether 5% constitutes a strong selection coefficient or not. I think it does. I also think this is consistent with our own perception of the mice on light and dark rocks. When we're out there working, the light mice on dark rocks stick out to the human eye more than the dark mice on light rocks. So I'm not surprised by this result. But I think it's a nice application of what you can do once you have the gene in hand. Well, after we published this, one question, of course, was whether this gene is responsible for color variation in other species. It was already known to be responsible in laboratory mice and a number of other domesticated species. But this led us to look next at lizards, and this was work done by Erica Rosenblum, who was a graduate student here in MVZ, and came to my lab. So she wrote to me and said, I'm working on these lizards that live on the white sands, and I said, well, you know, we have a gene. It would be worth looking at this. And so she came out and spent a month or so in my lab and looked at MC1R. And we didn't look in Scalopris. We looked in whiptails and earless lizards. And published a paper showing that there was an association between MC1R and color. And then more recently, she had this beautiful paper in PNAS in 2010, where they pursued this further and actually did some very nice functional tests showing that the alleles that we had inferred in this earlier paper to encode receptors that function differently. I thought this was pretty interesting that the same gene that was causing color variation in New Mexico in mice was also responsible for color variation in lizards in New Mexico. Hopi then followed this up looking at MC1R in some beach mice. So these beach forms, this is Paramiscus polianotus that lives on the barrier islands off the coast of Florida, where the soil is very light, and then the inland forms are darker, and also found that MC1R underlies some of the phenotypic difference in color there. And I'm going to come back to this at the end. So from now a lot of organisms, we know that gain-of-function mutations can produce dark color at this gene, including jaguars and skuas and mice and a bunch of domestic species. And loss-of-function mutations can produce light color or red color, including red hair in humans. So for the redheads in the crowd, you have more pheomelanin than the rest of us, and that's because of a mutation at MC1R. Now, in humans, it explains a relatively small proportion of the phenotypic variance. Nonetheless, it's a very well-established association. Many, many papers have been published in the last 8 years. I feel like we and Nick Mundy and Biff Birmingham published this first in Banana Quits, sort of opened this floodgate, and everybody and their brother, forgive the expression, went out and started sequencing MC1R. And sure enough, we see that in many different organisms, mutations in MC1R underlie color. Much has been made of this, sort of the remarkable fact that this gene is implicated in color variation in many species. I just want to say, I think that there is an enormous publication bias in this result. MC1R is an easy gene to study. It's got a single exon. It's 954 base pairs long. It's pretty easy to pull it out of a lot of different organisms. There are many other genes that affect color, and as I'll show you in a minute, some of those are implicated in the mice that we study. Nonetheless, this gene has been invoked and shown to be involved in associations with color variation in a wide variety of species. So MC1R is important in everything from lizards to mice, to pocket mice. What if you look within pocket mice now at these other populations? Is it MC1R in every case? And as I'll show you, the answer is most definitely not. And this is what we've been working on for the last 5 years, and we haven't published any of what I'm about to say because it's been very, very difficult to even get to where we are, and I want to publish a complete story from this, and I think we're very close, but as you'll see, it's technically been quite challenging. So just to remind you, here are the mice again. We're interested in asking, given that MC1R causes this difference, is MC1R responsible for dark color in those other populations? And we were able to answer very quickly that that was not the case just by sequencing the same gene in these other populations and showing that there was no variation. And so that ruled out there was no association upstream and downstream. That was an easy question to answer. And then left open the question of, well, what genes were involved? So we spent quite a bit of time trying to breed these mice in captivity. As a geneticist, my first thought was, well, let's just do some crosses. How hard can it be? Although I do remember learning from Jim Patton when I was an undergraduate here that heteromites are really tough to breed in captivity. Nonetheless, I set up many cages in my home and paired mice and tried to do everything. And I did it at home because I really wanted to sort of see and think about it. And I was completely unsuccessful. I spent 2 years trying to get them to cross. And then I said, well, look, they breed outside, let's just build an enclosure out in the desert. And so that's what we did. We went to New Mexico and studied Jim Brown's famous ecological study site where he has these beautiful enclosures, and we copied that design. So we built a fence. These mice hop and they burrow, so we built a fence that goes about 3 feet above ground, 3 feet below ground. It was very difficult work, but at a study site just a few miles from campus. So it was, for some reason, I'm down here building the fence. That's about. And I had more dark hair. Gabriela Vlasiak was a graduate student who really spearheaded this work. Here's a rattlesnake on the inside of the enclosure. The enclosure was 50 by 50 meters. And we did the most obvious, simplest genetic cross. This is this is the most difficult QTL mapping experiment I can imagine doing just because of the organism. But there are ways of working with non-model organisms, and I'm preaching to the choir here to this crowd to say that. But in this case, it paid off. So we took 6 light females from Arizona and put in 10 dark males from New Mexico. We chose animals from different populations because we wanted to ensure that there would be markers that were different between the populations so that we'd have something to track. We knew then that any breeding that came had to be from crosses between these animals. We marked all the animals. We trapped throughout the summer every other night, so we were able to see that all the mice were actually in the enclosure. We did that for 5 months or so. We set traps outside of the enclosure. Very rarely we caught some mice outside of the enclosure. Not often. What we found was the F1s were all light. Every single one of them, and we established through genotyping that they were all light, and this told us immediately that dark is recessive over light, and is consistent with the involvement of agouti or mahogany alleles in other species. Mahogany was that other gene I showed you right at the beginning. And then we did a back cross over the next 2 years. They don't have large litter sizes, but we took those F1 females and back crossed them to 2 dark males. Why only 2? In that first year when we had 10 males in the enclosure, a quarter hectare, 8 of those males were killed. So only 2 remained. And so that also told us something about the spatial distribution of males in maintaining territory. I never would have guessed that that would have happened. 2 dark males then sired all of the offspring crossed to this F1 female, and we got 12 light and 21 dark offspring. And even though those ratios look different with this sample size, they're not different, significantly different from a 50-50 ratio. They might be, but we can't say it with this sample size. At any rate, this is clearly consistent with simple Mendelian inheritance. We caught, in some cases, pregnant females that gave birth in a single litter to both light and dark animals, which we could bring them in the lab and let them give birth and let those animals grow up. And this is in juvenile pelage here, so they don't look quite as different, but showing without a doubt that these are the crosses. We thought we had genotypes on all of these animals. So dark is dominant in Arizona. This is MC1R I showed you before. Dark is recessive in New Mexico, and this suggested to us that agouti might be involved.
without going any further, I've shown you that the genetic architecture is different in these two populations. And so Haldane believed, of course, that most adaptive mutations were dominant. Here, that's clearly not the case. We have 1 and 1. The next thing was to take SNPs in our candidate genes and look for associations in this cross. And I'll just cut right to the chase and say we found a SNP in agouti that was different between the New Mexico and Arizona parents. All the F1s were heterozygous, and the back-cross offspring showed a perfect correlation between this SNP in agouti and that color difference. So we got that result 2 years ago, I think, and you would think, well, from this to, doesn't that just tell you agouti is involved? It's not as simple as that. We do think agouti is involved. Either a recessive mutation at agouti or at a nearby gene determines dorsal pelage. We couldn't at this point rule out the possibility that it was a linked gene, because in a cross, associations will extend over much longer distances. So one of the first things we had to do was look at nearby genes until we found a decay of this association, and we've now done that. What's been difficult is characterizing, what we're looking for is the causative mutation. I want to find a mutation that I have some confidence is actually causing this. The agouti gene is a monster. I mean, it's not unusual from the perspective of mammalian genes, but extends over 120 kb of genomic sequence. There are untranslated exons far upstream of the start codon, which is down here. And so we have laboriously been trying to walk through this whole area to characterize all of the mutations that are different between these. In the interests of time, I'm not going to go through all of those molecular details. I will say that I think that we're getting close, but I don't have a causative mutation to show you. At the same time, we went back to this other population, Kenzin, a different lava flow, and having now characterized the gene in great detail, we had many more SNPs to look at. And lo and behold, we found in this other population a very nice correlation between a SNP, a non-synonymous SNP in agouti and color. And in this population, we actually see 3 phenotypes. A dark, a sort of intermediate with a small band of pheomelanin on the hair, and a light phenotype. And it shows this very strong association. We see a decay of LD upstream and downstream, and so this really implicates this. But of greatest interest is that this haplotype in Kenzin that seems to be associated with dark color is different from the one that we see in Kerizoso. And what that means, I believe, is that the agouti gene is involved in dark color in both of these populations in New Mexico, but that it's a different mutation in each case. So we now have what I believe are 3 different origins of dark color, phenotypically very similar, but not identical. And after the fact, you can go back and look at these mice and notice subtle differences that maybe you didn't notice as much at first. MC1R clearly in this case, we think agouti in these other 2 cases, but different mutations. Okay, the last thing I wanted to say about melanism has to do with the genetic architecture of adaptation. And this is not something I have said in print, or Hopi Hoekstra has said in print anywhere, but for me it's really one of the more striking things that's come out of looking at, in this case, MC1R in different species. So I made the point simply that MC1R is involved in color variation in different species, but in a very different way. In pocket mice, it's basically a Mendelian trait. If you have this dark haplotype, you have a dark mouse, and if you don't, you have a light mouse, and that's the end of the story. It accounts for at least 90% of the phenotypic variance. We've taken reflectance measurements on all of these mice. In the beach mice, MC1R accounts for about 10% of the phenotypic variance. If you look just on the rostrum of these mice, there it accounts for 30% of the phenotypic variance. But if you look on the back, this, what to me at least looks like sort of one of the more noticeable differences between these mice, it's accounting for a very small amount of the phenotypic variance. So here we have 2 species of rodents that are both under the same kind of selection pressure, that is selection for crypsis. We have the same gene involved, and yet the genetic architecture is very different. And so the question is, why Why is that? Why would the genetic architecture of these traits be so different? And in some sense, I think the answer might be fairly straightforward. And it's easiest to think about in terms of Fisher's geometric model of adaptation. If you imagine an organism is out here on the perimeter of this circle, and these axes are some measure of phenotypic space, so maybe this is hue and this is reflectance, for example. And the optimum is here. So imagine this is a light mouse that's landed on a lava flow, and this point in the middle is the ideal color to match the lava. Then a mutation of very large effect may be beneficial. It will take you close to the optimum. So Fisher pointed out many years ago that mutations of large effect are likely to be beneficial when you're far from the optimum, but as you get closer, this circle gets smaller and smaller, and so successive mutations are expected to be of smaller and smaller effect. Those that are big, once you're closer to the optimum, are going to overshoot, and now the fitness is lower. So the closer you are to the origin there, the higher your fitness. And so mutations of large effect may be beneficial when you're far from the optimum. Mutations of large effect are likely to be detrimental when the organism is close to the optimum. Well, if we now go back to the environment, in the mice that we've been studying, in the pocket mice, in just about every case, the environment is very coarse. That is, there's a spot in the ground which, if you cross, you go from a light substrate to a dark substrate. And so when these lava flows originated, my guess is that the mice that first colonized them were really light mice that landed on very dark rocks. And a mutation of large effect is likely to be favored. In contrast, the beach mice, Peromyscus polyanotus, if you look at the sands there, there's a more continual gradation from light to dark. And a mouse that moved into a new habitat was probably faced with a relatively small change in color, and a mutation of large effect then would overshoot. And so I think that the coarseness of the environment may dictate the genetic architecture of traits, perhaps in a more general way. I didn't talk about this at all. We also looked in gophers a number of years ago, and we didn't see any association between color and MC1R variation. Although we didn't have a lot of power to detect small effects. What we were able to rule out was the possibility of a large effect. Well, gophers show a fairly nearly continuous gradation of colors, and it may be that they're more like beach mice, or it may be that MC1R is not involved. So that's what I wanted to tell you about adaptive melanism, and then after I summarize here, I'll just take a couple of minutes to tell you about this new project that we're starting. I've told you that a single gene of major effect, MC1R, causes melanism in one population, and it's due to gain-of-function dominant mutations that are in the structural regions, not regulatory regions of the gene. So here I'm touching on many of those questions that I raised at the outset. Selection on mismatched alleles is strong and asymmetric. It's bad to be a light mouse on a dark background, and probably worse than to be a dark mouse on a light background. MC1R is also responsible for adaptive color variation in a wide range of vertebrates. I think this is interesting. I also think there's a publication bias here, and so you should remain open to the possibility that there may be many other genes, and as it becomes easier to characterize genes, we'll see some of this variation. Despite the fact that MC1R causes color variation in lizards and mice in New Mexico, different populations of mice in New Mexico have nearly identical melanic phenotypes that have arisen independently due to changes at different genes or different changes at the same genes. So we have now what I believe are 3 different genetic solutions to the same evolutionary problem, and they include both dominant and recessive and sort of a co-dominant trait. And finally, the graininess of the environment, as I said, I think might dictate the genetic architecture of traits. Well, this has been really fun work to do. It's just, I've loved it because of its sort of simplicity, and it's very clear, and it served as a great example. But it's, in some sense, it's kind of low-hanging fruit. We used a candidate gene approach, and I think that a lot of phenotypic variation is going to be quantitative, and a lot of the phenotypic variation that we're really interested in is continuous. And so this led me to think about what would be interesting to study moving to the sort of next level of complexity. And I think that environmental adaptation in house mice provides a great opportunity. So Mus domesticus has been introduced into the Americas in the last few hundred years. They live in a wide range of environments, from the equator to extreme latitudes, in both North and South America, from sea level to over 3,000 meters. And these populations show evidence of adaptation to different environments for complex traits, including things like body size and behavior. Now, probably many of you are familiar with the beautiful work that's been done by a number of people in Drosophila, showing that there are clines for things like wing size and ADH alleles and a whole bunch of things over wide swaths of latitude that are replicated in, say, North America and Australia, but in reverse. So there are clines for ADH alleles along the eastern seaboard of North America and Drosophila melanogaster, and you see the same thing in Australia, but in reverse orientation. And this sort of replicated pattern, like the lava flows, provides, I think, convincing evidence for selection. And I thought, well, mice might provide another context for looking at that. And so if you were to scan the genome and look for changes across the genome that are similar as you go from Ecuador to Ushuaia in southern South America and from Ecuador to Alaska, you might have some confidence that those reflect selection in response to, or changes in response to similar selection pressures. This is some work that motivates it, and this was done by Carol Lynch and published in AmNat in 1992, and I just think it's gorgeous work. What she did, and there's not been any follow-up to this, is sample house mouse populations from Florida to Maine. And she measured body weight, and what she found was they follow Bergman's rule. The mice from the north are bigger than the mice from the south. She also found that they make bigger nests. If mice from Maine make bigger nests. Most interestingly, she brought them into the lab and showed that the heritability of these traits was high and that differences persisted over 3 generations. So not only are they showing changes that we think correlate well with latitude and might reflect adaptation to different climates, but they have a genetic basis. So this is evolution that's happened over the last couple hundred years. So what we are planning to do, in fact, we've started this, but we're not very far along, is to sample populations every 7 degrees of latitude from Ushuaia to Alaska. So I did the first collecting trip this past January to Ushuaia in the middle of summer. We planned by having this very large latitudinal gradient to be able to compare changes from equator to north and equator to south. We're also doing paired transects of altitude in 2 different places in the Andes, one in Ecuador and one in Peru, from sea level to over 3,000 meters. We have a nice SNP chip we can use to genotype 600,000 SNPs. We're doing this on hundreds of mice, so we have markers for every single gene and non-coding region of the genome. And so we can look for genomic regions showing signatures of adaptation. I was about to say one other thing that was relevant, and it just went out of my head, so I'm if it pops back in, I'll tell you. Ultimately, what we would like to do, though, is make links between genotype and phenotype. So in addition to what I've described, we're going to establish 16 new inbred strains from these extreme environments, that is, bring wild mice into the lab, breed them, so that we can phenotype them for many, many different physiological and metabolic traits. And then if we have an allele at a particular location that we think shows evidence of parallel changes, we will have those mice and we can actually do crosses and try to see if those traits track in crosses. And then we plan to sequence the genomes of all of those strains, so we will actually have a complete characterization of the alleles. One of the things I was going to say is that this sort of thing, minus bringing them into the lab, has been done recently in humans. And there are strong associations in humans between all sorts of climatic variables and genes underlying metabolic traits, including those important for metabolic disease. And so there's a huge interest in the human genetics community for mouse models that will be useful for studying this. With mice, of course, we can bring them into the lab and then hopefully get at the genetic basis. Conceptually, for me, this is not as different from studying the genetics of melanism on different lava flows. It has a lot of the same attractions. We have, I like the idea of natural evolutionary experiments. So the lava flows were always attractive to me because they were replicate lava flows, and we could study the same process on different lava flows. We can kind of do the same thing here with replicate transects. And I think that's a convincing way of looking for adaptation. The other thing that we can do, though, is by looking in the lab, we have actual alleles and strains that we can go back to. And unlike the study of melanism, the goal here is to really use this approach to start to understand the genetic basis of complex traits. My long-term goal, let's say in 5 years we do all this and we have 10 genes that we think are really important for high-altitude adaptation in mice. Well, wouldn't it be interesting to then take those same genes and look at an Acodon species that lives at high elevation in the Altiplano and ask whether we can use that mouse model to sort of get into the biology of species that have been there for many generations. So that's more of the long-term goal. With that, I will thank the people who really did a lot of the work that I've talked about. Megan Pfeiffer-Rixey is a current postdoc in the lab, and Jillian Moore is an undergraduate, Gabriella, a graduate student, and they really did a lot of the coat color work. Earlier, Jim Prince, a former technician, Hopi Hoekstra, postdoc, and these 3 undergraduates all contributed. A lot of trapping was involved in this work. And, I have these people to thank, and both NSF and NIH provided support. So I will stop there and thank you for your attention and take any questions.
I know it's after 1, and some people will probably have to run off to classes or other commitments, so why don't we just take a moment to let people